Hello friend, welcome to another video tutorial, and today we will discuss about the structure of anther. As we know, the male reproductive organ of a flower is known as androecium, and androecium is consist of three structures named as anther, filament, and connective. If we cut a section from this middle of this anther, we found that there is this structure like this where there is lobed. Mostly angiospermic anther are bilobed, and each lobe consists of two microsporangium, two microsporangium, or either they are also known as later on when they mature pollen sacs. Okay, known as pollen sac. The TS of anther shows that it have an outermost layer which is known as exothecium, which is known as exothecium. Just inside to this exothecium, another layer of tissues is present, which is known as endothecium. And just inside to this endothecium, another layer is also present, which is known as middle layer. Middle layer. Inside this middle layer, another layer of multiple layer of tissues is present. This layer have the different type of cells which looks like amoeboidal. These cells looked like amoeboidal and covered the sporogenous tissues. And this layer of tissues who have more than one nuclei is known as tapetum. Its main function to nourish the pollen grains. And inside this tapetum layer, a mass of tissues is present which are known as sporogenous tissues. Sporogenous tissues, and each sporogenous tissues have capability to convert into microspore. The cells of tapetum have many functions, like it produce auxin hormone, which increase the rate of cell division and increase the microspore formation. Similarly, it produce obese bodies. These obese bodies are filled with sporopollenin, and this sporopollenin produce the exine of pollen grain. The outermost layer of pollen grain is known as exine, which is made up of a sporopollenin, and the sporopollenin is the toughest organic molecule which cannot be digested by enzyme. pH like acidic and alkaline. It can tolerate the temperature above 300 degrees Celsius and can sustain in freezing temperature too. So these are produced by tapetum. Another function of tapetum is to produce pollen kit, pollen kit, a sticky layer present on pollen grain surface in animal pollinated plants. Similarly, pollen except that tapetum also produce callase enzyme which digest callose, and the callose is present in between the which get together the microspore microspore tetrad. So this callase enzyme digest this callose and separate the microspores. So these microspores further divide and developed into pollen grains. Okay, so that is here a mass of tissues is also present which is known as connective. It absorbs water and nutrients from this filament, and this filament absorbs water and minerals from the stem. So it provides nutrition to all cells of the anther. I hope you understand the structure of anther. The structure of anther consists of four layers, namely outermost layer, which is also known as epidermis, exothecium. Inner to this exothecium, another layer which is known as endothecium. Inside this endothecium, another layer is known as middle layer, and below this middle layer, a amoeboidal multinucleate layer is present, which is known as tapetum. And inside this tapetum, a mass of cells is filled up, which is known as sporogenous tissues. These sporogenous tissues further developed into microspores, and this is all about the structure of anther. How one more thing is there. In some plants, there is only one lobe, so there is only two pollen sacs are present. In some plants, there is two lobe, there is four pollen sacs are present. Thank you so much for your time.